In this video, we will talk about the human mitochondrial DNA and examples of mitochondrial inheritance. So, let's discuss human mitochondrial DNA. First, human mitochondrial DNA has 22 transfer RNA genes, 2 ribosomal RNA genes, and 13 protein coding regions. 14 of the transfer RNA genes are expressed in the clockwise direction, and 8 are read counterclockwise. Animal mitochondrial DNA is extremely compact. There are extensive differences in the detailed gene organization found in different animal taxonomic groups, but the general principle of a small genome encoding a restricted number of functions is maintained. In mammalian mitochondria, the genome is particularly compact. There are no introns, some genes actually overlap, and almost every base pair can be assigned to a gene. With the exception of the D loop, it is a region involved with the initiation of DNA replication. Mitochondrial DNA has 16,569 base pairs. The complete nucleotide sequences of animal mitochondrial genomes show extensive homology in organization. There are 13 protein coding regions. All of the proteins are components of the electron transfer system of cellular respiration. These include cytochrome B, 3 subunits of cytochrome oxidase, 1 of the subunits of ATPase, and 7 subunits or associated proteins of NADH dehydrogenase. Moving into detailed structure of human mitochondrial DNA, or human mitochondrial genome. Mitochondria are semi-autonomous replicating entities with their own genetic apparatus. Mitochondrial DNA, which has been called the 25th chromosome by some, and chromosome M by others. It is circular, with 16,568 base pairs. Mitochondrial DNA carries the genetic information for 2 ribosomal RNAs, 22 transfer RNAs, and 13 proteins. The complete nucleotide sequence of human mitochondrial DNA is known. The 13 proteins encoded by the mitochondrial genome and translated within the matrix of the mitochondria are involved in oxidative phosphorylation functions. This all has been already discussed. 7 of these mitochondrial proteins, mitochondrially encoded NADH dehydrogenase 1, mitochondrially encoded NADH dehydrogenase 2, mitochondrially encoded NADH dehydrogenase 3, mitochondrially encoded NADH dehydrogenase 4, mitochondrially encoded NADH dehydrogenase 4 lighters, mitochondrially encoded NADH dehydrogenase 5, mitochondrially encoded NADH dehydrogenase 6 are subunits of complex 1 of the electron transport chain. 1 mitochondrial cytochrome B of complex 3, 3 cytochrome oxidase 1, cytochrome oxidase 2, cytochrome oxidase 3 of complex 4, and 2 ATPase 6 or A6, ATPase 8 or A8 of complex 5. Nuclear genes encode about 70 additional proteins that make up the oxidative phosphorylation system, and about 1,000 other mitochondrial proteins promote replication, transcription, and translation, facilitate the transport of various compounds through mitochondrial membranes, and metabolize fatty acids and pyruvate. Let's move into more details of human mitochondrial genome. The nucleotide positions numbers are marked at various intervals in a counterclockwise direction. The transfer RNA loci are indicated by single-letter amino acid codes: phenylalanine, proline, threonine, glutamate, leucine, serine, threonine, arginine, glycine, lysine, aspartate, serine, tyrosine, cysteine, asparagine, alanine, tryptophan, methionine, glutamine, isoleucine, leucine, and valine. And the ribosomal RNA sites are 12S and 16S. Now let's discuss differences between the universal and human mitochondrial genetic codes. Well, UGA is a stop codon, but in mitochondrial DNA, it codes for tryptophan. IgG codes for arginine, but in mitochondrial DNA, it is a stop codon. IgA codes for arginine, but in mitochondrial DNA, it is a stop codon. IUA codes for isoleucine, but in mitochondrial DNA, it codes for methionine. IUU codes for isoleucine, but in mitochondrial DNA, it codes for isoleucine elongation and methionine initiation. Moving forward, in many organisms, organelles and their DNA are inherited from one parent. For many eukaryotic species, and particularly in animals, organelles are inherited uniparentally. When uniparental inheritance occurs, progeny most often inherit their organelles from the maternal parent, but many exceptions exist. For example, in bananas, inheritance of the chloroplast genome is maternal, while that of mitochondrial genomes is paternal. The situation is reversed in alfalfa, while in sequoia trees, both chloroplast and mitochondrial DNAs are paternally inherited. We focus here on one of the classic examples of the more common maternal pattern. Let's discuss example 1 of maternal inheritance. So, maternal inheritance of the Pocky phenotype in Neurospora, in the octads from a mating of air, Pocky, positive, which has blue cytoplasm and red nuclei, female gamete, and a normal, negative, having brown cytoplasm and yellow nuclei, male gamete, and then matting takes place. That leads to the production of half the spores that are negative and half are positive. All of the spores are Pocky, because this phenotype is controlled by a mitochondrial gene, and all the mitochondria are supplied by the larger female gamete, as female parent is Pocky. So, offspring are Pocky. In other case, which is reciprocal of first case, in which a normal, negative, having brown cytoplasm and yellow nuclei, female gamete, and other POCI, at positive, which has blue cytoplasm, and red nuclei, male gamete, and then natting takes place. That leads to the production of, give rise to, half normal, add negative, and half normal, add positive spores. However, all the spores are normal, not POCI, because this phenotype is controlled by a mitochondrial gene, and all the mitochondria are supplied by the larger female gamete, as female parent is normal, so offspring are normal. Now, let's discuss second example of maternal inheritance. In maternal effect, also referred to as maternal influence, an offspring's phenotype for a particular trait is under the control of nuclear gene, products present in the egg. This is in contrast to biparental inheritance, where both parents transmit information on genes in the nucleus that determines the offspring's phenotype. Both of these concepts are well explained in our previous videos. So, let's discuss it using second example. Lymnaea coiling shell. Lymnaea peregra is an excellent example of maternal effect. On a permanent rather than a transitory phenotype, some strains of this snail have left-handed or sinistrally coiled shells, having small d and small d, while others have right-handed or dextrally coiled shells, having capital D and capital D, or capital D and small d. These snails are hermaphroditic and may undergo either cross or self-fertilization, providing a variety of types of matings. Let's discuss it. Now, if a matting occurs between parents, such as mother, which is purely dextral, and father, which is sinistral, these gametes will be generated from mother, and these gametes will be generated from father, as a result of which, in F1 generation, all offsprings have capital D and small d. So, all offspring will be dextral, as capital D is dominant and it is from mother. In next fertilization, between F1 generation, both of which are dextral, these are gametes generated from mother, and these from father. So it leads to production of F2 generation. There should be 3 dextral, and 1 should be sinistral, as it has small d and small d, but it is also dextral instead of sinistral, because of maternal effect. So, a maternal effect is evident in this offspring, where the genotype of the maternal parent, rather than the offspring's own genotype, controls the phenotype of the offspring. Now, let's discuss the genetic risk assessment. So, the genetic risk associated with a mitochondrial disorder is that: example 1, the affected mother and normal father. There is a 100% chance of having an affected daughter with a range of phenotypes, and there is a 100% chance of having an affected son with a range of phenotypes. But in example 2, normal mother and affected father, there is a 0% chance of having an affected daughter. There is a 0% chance of having an affected son. So this was all. In our next video, we will talk about the genetic disorders involving mitochondria and therapy.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.